So this is about where we left off last time. We were talking about mitosis and meiosis, and really comparing the two. So remember, like really broad brushstrokes, mitosis is a process of nuclear division where you end up with two daughter cells that are genetically identical to each other. And that's the process that we use for growth and development. When we're developing as embryos, all those cellular divisions are accompanied by a mitotic nuclear division, right? Meiosis is used fundamentally differently. This is the process that we use to create gametes. That is, this is the nuclear division that's associated with gamete production. And it is very much like mitosis, but instead of ending up with two daughter cells that are genetically identical to each other and to the mother cell, you end up with four daughter cells that are genetically distinct from each other and genetically distinct from that original mother cell. Additionally, each of those four daughter cells and daughter nuclei have half the genetic complement, right? So they start off with 46 chromosomes, each ends up with 23 chromosomes. Now, the phases of meiosis and mitosis are the same. There is prophase, metaphase, anaphase, and telophase. But we're going to do those twice in meiosis. And really, they're very similar to each other, right? There are some differences between meiosis 1 and mitosis, but meiosis 2 and mitosis will be nearly identical to each other. So we started last by making a list of the differences in divisions between meiosis and mitosis. And the first of which is, during prophase 1, there's something called synapsis that occurs. And this is where homologous chromosomes pair up, right? So you'll remember that in prophase of mitosis, those chromosomes began condensing and the mitotic spindle was attaching to them, and it was kind of pushing all of them in a single line towards the center of the cell. But in prophase 1, things happen a little bit differently. Instead of all of all the chromosomes aligning towards the center, you're going to have homologous chromosomes that pair, and then those pairs get pushed towards the center of the cell, right? So that looked something like this, where we had a cell, and then in that cell we had homologous chromosomes, and we'll use blue for mom's chromosomes and red for dad's chromosomes, that are starting to pair up, right? And what eventually is going to happen is the mitotic spindle, and it's still called the mitotic spindle. The mitotic spindle that's forming at the poles of the cell will be attaching, ultimately, to those homologous pairs. Now there is a hard and fast connection between these chromosomes. And it's a hard and fast connection that's facilitated by some proteins called cohesins. It's the same proteins that hold sister chromatids together. So this mitotic spindle is going to grow out and attach like this. That's synapsis, where homologous chromosomes pair up. And remember, a homologous chromosome are two chromosomes that have the same genes on them. What the genes look like, the alleles that we haven't really talked about yet, might be a little bit different. But basically what happens, right, is the chromosome that carries eye color from mom pairs up with the chromosome that carries eye color from dad, right? And they all line up towards the center of the cell. And that's what happens in metaphase 1. Instead of all of the chromosomes aligning in a single line at the center of the cell, we get homologous chromosomes that pair up and are aligned at the center of the cell. So this is prophase 1. And in metaphase 1, what happens is those chromosomes are completely condensed at this point, and they are aligned at the center of the cell in pairs. Like so. And then we still have those other microtubules that grow out from the centrioles, right, that act kind of as anchors that attach to each other. So this is metaphase 1. And And then the phase that I believe we ended up talking about was anaphase 1. You'll recall in anaphase of mitosis, what happens is sister chromatids get pulled towards opposite ends of the cell. Right? Those cohesins that are holding sister chromatids together are cloven. They're broken apart. And then those sister chromatids migrate. In anaphase 1 of meiosis, however, it's the cohesins that are holding homologous chromosomes together. They get broken apart. And chromosomes migrate towards opposite ends of the cell. So anaphase 1 would look something like this. Okay, so homologous chromosomes have now separated from each other and they're at opposite ends of the cell. Very different than anaphase of mitosis, right? Because it's not sister chromatids that are separating, it's homologous chromosomes that are separating. You guys, I can almost guarantee you that as a written item on the exam, you will be asked to compare mitosis and meiosis. So these differences that I'm outlining aren't trivial. You'll definitely be required to know them. Sorry, I forgot to label this one. This is anaphase 1.
If you drew it, would it be okay? Or
Yeah, for sure.
put some word? Okay.
Yeah, just as long as you kind of label things so I know
Okay.
what you're referring to. Okay, so at this point, how many chromosomes are in that cell in anaphase 1? 4. And how many chromosomes did we start off with? 4. So nothing has happened to the number of chromosomes just yet. That changes between the next couple of stages. The last stage in meiosis 1 is telophase 1. And in telophase 1, we get the mitotic spindle that generally detaches from homologous chromosomes, and we might even get like nuclei reforming around those homologous chromosomes that are separated from each other. So it's kind of the opposite of prophase 1, right? Mitotic spindle detaches, and then we're going to get cytokinesis. So telophase 1 could look something like this. Sorry, it's a smaller cell. I didn't leave myself much room. Telophase 1, where we have homologous chromosomes that might even begin to unwind a little bit as well, that are at opposite ends of the cell from each other. For mom's or dad's chromosomes. I look something like that. And then we're going to get cytokinesis. So after telophase 1, cytokinesis, at least in animal cells, this is how it looks. Cytokinesis begins to occur where we get that cleavage furrow in between the daughter nuclei. So the last difference that I'm going to point out right now is that after telophase 1, cells become haploid. So it's important to remember that this occurs after telophase 1. So I have a diagram that's going to show you all that's happened in prophase 1 through telophase 1. So in prophase 1, we start off right. Homologous chromosomes begin pairing. This is called synapsis.
This is important because it facilitates crossing over. We talked a little bit about that last time, where we get homologous chromosomes that will actually exchange some genetic material. The leg of dad's chromosome, right, will flop over on the leg of mom's chromosome, will physically break and exchange those legs, so that dad's chromosome now has one of mom's legs on it, and mom's chromosome has dad's leg on it. That's facilitated by synapsis. And that process, again, is called crossing over. In metaphase 1, those homologous chromosomes have aligned at the center of the cell. And then in anaphase 1, homologous chromosomes are pulled apart from one another. And what I hope you can see in this diagram, though it's pretty far and a little bit obscured, is these chromosomes at this point aren't either entirely purple, nor are they entirely green. There is a purple chromosome with some green on it, and green chromosomes with some purple on it. That's the product of crossing over. And then in telophase 1, those chromosomes have migrated to opposite ends of the cell. And then we end up with, after telophase 1, in cytokinesis, 2 daughter cells that now have half the genetic complement. Yep?
Does crossing over happen in anaphase?
No, crossing over happens in prophase.
Okay.
Yep. Okay. Yes, Sarai.
So like how we drew it on there, like you colored it perfectly, it's not, that doesn't mean anything, right?
No.
Okay.
Yeah, that was just to illustrate chromosome movement.
Okay.
Okay, other questions? Okay. So after meiosis 1 is completed and cytokinesis has ended, then we go through meiosis 2. I'm not going to write these up on the board. I'm just going to go through them, and I'm going to move through it rather quickly because, like I said, this is identical to mitosis. So the first stage of meiosis 2 is prophase 2. And in prophase 2, if chromosomes have begun to unwind, and if nuclei have formed, all that stuff is undone. The nuclear envelope begins to break apart, and chromosomes begin to wind up really, really tightly. And then the mitotic spindle begins to grow out and attach to those chromosomes. Right? Exactly like what happens in prophase of mitosis. In metaphase 2, chromosomes have aligned at the center of the cell. And this is a true chromosome alignment. It looks exactly like metaphase of mitosis. Where if this is one cell, right, those 2 blue chromosomes are just going to be lined up right on top of each other. And the mitotic spindle is going to be attached to both chromosomes from both sides. In anaphase 2, what happens is sister chromatids are pulled apart from each other. Those cohesins that are holding sister chromatids together get unzipped or cloven, and then those sister chromatids migrate to the opposite ends of the pole, opposite ends of the cell. And then in telophase 2, we get the undoing of prophase 2, right? So the mitotic spindle detaches, chromosomes begin to unwind, and a new nuclear envelope forms around each of those homologous sister chromatids. Okay, so is it okay if I erase this? Does anyone want to take a picture of that before I erase it? So we'll follow the trajectory of just one of these daughter cells that we've created. In the beginning of meiosis 2, we'll make it mom's chromosome cells. What we have, right, is a nucleus that's formed around 2 chromosomes, and perhaps it begin to unwind a little bit like that. So that's the beginning. That's prophase 2. And remember, at this point, those blue chromosomes aren't going to be entirely blue, right? They're going to have some of dad's genetic material on them. They're going to be recombinant. That's what we call it. And then by the end, we're gonna have two daughter cells after telophase 2 that look something like this. And this is after telophase 2. So at meiosis 2, starting off in prophase 2, how many chromosomes did that cell have? 2. It only had 2, right? And then after telophase 2, how many chromosomes does each of those cells have? It's 2. It's 2, right? Because this is a chromosome, just like this is a chromosome. The only difference between this chromosome and this one is that this one has already been through the cell cycle and has 2 copies of all of its DNA. So meiosis 2 is the same as metaphase, or excuse me, is the same as mitosis. You end up with 2 daughter cells that have the same number of chromosomes. The difference, of course, is this one has 2 copies of all of the DNA, and this one only has 1 copy of all of the DNA. But that's okay, because those will go through the cell cycle, and their DNA will be replicated. Okay, so what questions have you about meiosis 2? I know I went through that quickly. Yeah, Carly.
So, after telophase, when the daughter cells have 2 chromosomes, are they then considered haploid because they have only half?
Good question. They're considered haploid at this point because they have half the number of chromosomes. It's not genetic material, it's number of chromosomes. We started off with a cell that had 4 chromosomes. And after telophase 1, they became haploid because they only have 2 chromosomes. Does that idea bother anyone? Is that confusing in any way? Please, you guys, think of it in terms of number of chromosomes, not amount of genetic material. Because we have half the genetic material, right? There's twice as